Hello, and welcome to another episode of Mad Scientist Brewing Beer Education Series. Today's episode looks at the process of how brewers propagate yeast to be used for the brewing process. It is typical to collect yeast from an active fermentation to be used for a subsequent batch of beer. The way this is done depends on the type of fermenter. Open fermentation vessels utilize the top cropping method, which has been used for centuries, in which foam produced by vigorous fermentation is skimmed off. This foam contains lots of healthy yeast cells which can be stored and reused. Another method for modern cylinder conical fermenters is to collect yeast from the bottom port, after first allowing trub and dead yeast to be removed. In either case, this healthy yeast is stored cold and ideally used fresh in a subsequent batch at the correct pitch rate for the type of beer being used. In many cases, a fresh batch of yeast is added to wort rather than collecting an old batch. This may be done when the current yeast has run through so many generations that it may affect quality, or when there is need for a different variety of yeast. Whatever the case, it is necessary to produce enough yeast to ferment the often very large batches of beer, and it all starts with a few yeast cells. Breweries will often keep a library of yeast which is stored cold to prevent reproduction and possible mutations. Yeast is stored long-term in vials called slants, which contain nutrient-rich laboratory agar, which has been allowed to set at an angle in the tube to maximize surface area. Previously cultured yeast cells are spread on the agar aseptically and stored cold, sometimes under mineral oil to keep out oxygen, and sometimes even kept frozen to prolong storage life. When the time comes to propagate the yeast, the slants are taken out of cold storage and the process of scaling up the cells begins. This is done with subsequently larger batches of wort, which are used to feed the yeast, and cause them to multiply until the target cell count is achieved. This graphic illustrates the process of scaling up the yeast. The initial cells are added to a small volume of wort at a relatively low specific gravity, about 1.020, so as not to shock the yeast so much. This is allowed to ferment for 1 to 2 days to double the amount of cells. The fermenting beer is then added to fresh wort, this time with gravity closer to 1.040, such that the volume is 10 times the original. This is allowed to ferment another 1 to 2 days before scaling up again. Eventually, the size will exceed what is suitable for the laboratory and move to a piece of equipment called a Carlsberg flask, which is a sealed chamber used for propagating the yeast. Depending on the size of the brewery, further scale-ups may require larger tanks, the size of some small fermenters, to continue propagation. These steps continue until the target yeast count is obtained, which is monitored by brewing scientists throughout the process by pulling samples to count the number of cells per milliliter of wort, and also how viable these cells are. Although more sophisticated these days, the process of growing and repurposing yeast has been done in one way or another for a few centuries. That concludes this episode on the topic of yeast propagation, and until next time, cheers.